Hello, my beautiful biologist. It's time for another biology with Miss Judd. Our information molecule is DNA. It stores instructions for making other molecules called proteins. Each gene stores the directions for making protein fragments, whole proteins, or specific proteins. DNA is so well suited to perform this biological function because of its structure. The development of a series of high performing enzymes are so fine tuned to interact with the molecular structure of DNA in super specific ways. The match between DNA structure and the activities of these enzymes are so effective and well defined that DNA has become, over evolutionary time, the universal information storage molecule for all forms of life. Nature has yet to find a better solution for DNA for storing, expressing, and passing along instructions for making proteins. In order to understand the biological function of DNA, first we need to understand its molecular structure. Sounds like it's time to talk to Miss Judd in the corner. Thank you, Miss Judd. Happy to be here to talk about Unit 4, Lesson 3, DNA structure and replication. Now, before we get through the functions of DNA, we need to understand the basis. DNA stands for deoxyribonucleic acid. DNA is the genetic material responsible for all traits of living organisms. In our eukaryotic cells, we have our organelle, the nucleus, and that's where we can find DNA. It is safe inside that nucleus. DNA is a nucleic acid. Nucleic acids are made up of building blocks called nucleotides. The DNA nucleotide is joined into long strands or chains by covalent bonds. It has three parts: the nitrogenous base, the phosphate group, and deoxyribose sugar. And I actually want you to draw and label one nucleotide. DNA is made up of nucleotides and long strands or chains held together by covalent bonds. So we have building blocks. Here is one nucleotide, and it's held together with a covalent bond. If you notice, this covalent bond is in between a phosphate and a sugar. This creates a sugar-phosphate backbone. Covalent bonds are super strong, and it holds the sides together so that DNA doesn't break easily. We have our bases sticking out to the side. Our nitrogenous bases are the part of the genetic information that is the code. The sugar and phosphate never change. It's deoxyribose and it's phosphate. The nitrogenous base is the part of the nucleotide that has that genetic information. The order of the A's, T's, C's, and G's is where we get our code to build different things. The four bases found in DNA are adenine, guanine, cytosine, and thymine. We have multiple scientists that have contributed to our understanding of DNA and its function and its structure. Edwin Chargaff carried out a lot of different biochemical studies and discovered that the percentage of adenine, A, is equal to the percentage of thymine, T, in almost any sample of DNA. The same thing is true for other nucleotides. Guanine is the same and equal to cytosine. We call this Chargaff's rule, where A equals T and C equals G. You'll always find A paired with T and G paired with C. Next, we have one of my favorite. In the 1950s, Rosalind Franklin used a technique called X-ray diffraction to study the structure of DNA. She took a huge amount of DNA and then stretched the fibers to make them parallel. She then aimed an X-ray beam at the sample and recorded the scattering pattern that was made on the film. Her results showed some very important clues to DNA structure. The X-shaped pattern that we see here shows that DNA is twisted around each other like the coils of a spring, known as a helix. The angle of the X suggests that the two strands of the molecule. Other clues suggested that the nitrogenous bases of the nucleotides were on the inside of the molecule. Rosalind Franklin laid the groundwork for Watson and Crick. They approached the problem of DNA structure by building three-dimensional models made of cardboard and wire that would be possible to form a biochemical point of view. They twisted and stretched the models in various ways, but their best efforts did nothing to really explain DNA's properties. When they found Franklin's work in 1953, they realized how they needed to fix their model. Franklin's X-ray pattern allowed Watson and Crick to build a model to explain the specific structure and properties of DNA. They twisted the double helix model to explain Chargaff's rule of base pairing and how the two strands of DNA are held together. So we now have DNA that is a double helix with the two strands of nucleotides wound together with my bases paired in the center. It looks something like this. G is always bound to C, A is always bound to T. This arrangement of these strands run in opposite directions. This arrangement is called anti-parallel. It means that the nitrogenous bases on both strands come into contact at the center. So I have one strand going up and one strand coming down, which means my bases meet in the middle. Now, to make sure that my two strands meet in the middle and stay connected, the double helix is held together by hydrogen bonds. The hydrogen bonds a bond between adenine and thymine and guanine and cytosine. That's why Chargaff saw the same percentage of each. The two strands of a DNA molecule join at these bases. Remember that the sugar and phosphate are held together by covalent bonds. The strands are held together at the bases by hydrogen bonds here. Hi team, Ghost Miss Judd here to kind of interrupt. Now that we understand DNA as a molecular structure, being a double helix, having two complementary strands that twist around each other, carrying genetic information within its sequence of the nucleotide nitrogenous bases, now we can jump into the super exciting world of replication. DNA replication is where the DNA molecule is copied into two identical copies, making sure that each new cell receives a complete set. This is how we pass on our genetic information, especially in mitosis. It's so important. All right, back to Miss Judd in the corner. To understand DNA replication, we want to look at the big picture, the who, the what, the when, the where, and the why. So all living cells go through this process because all living cells have DNA. This is where we copy all of the cell's DNA. If you remember from our mitosis conversation, in interphase, there's an S phase where DNA is synthesized. This is that DNA synthesis. This is what happens before a cell divides in the cell cycle. In eukaryotes, this exists in the nucleus of the cell because that's where DNA is kept and kept safe. The whole purpose of this is to pass on information needed to run a cell. We have 4 steps for this process. First, DNA is unzipped at the replication fork by an enzyme called DNA helicase. Hydrogen bonds between those nitrogenous bases are broken so that the molecule can open up. This leaves the nitrogenous bases available and serve as a template. So step 2, DNA polymerase adds in new complementary nucleotides to each side. We use the base pairing rule here to build a strand of DNA that is complementary to the original template strand. A bonds with T, C bonds with G. DNA polymerase, this enzyme that adds in these nucleotides, also proofreads and fixes errors. Finally, the two identical new strands separate. To put it all together, we have this big picture of DNA replication. Our DNA molecule is opening at my replication fork. I have DNA polymerase on each side of the molecule. Because of the anti-parallel, polymerase moves in opposite directions. New nucleotides are added by the DNA polymerase molecule. We call this process semi-conservative replication because each daughter strand is made of one old and one new strand. We always use an old DNA side as a template to make sure that the copy is exact. We want to make sure that we are making an exact copy that is identical. That brings us to an end of Unit 4, Lesson 3. Let's send it back to Miss Judd. Thanks, Miss Judd in the corner. That was so helpful. You guys, you're doing such good work. Please make sure you bring these notes to class so that we can work with it some more. Have such a good day and make good choices.